Good afternoon, and welcome to today's webinar, Environmental Chemicals and Breast Cancer. My name is Michelle Dixon, and I am the Program Director for the Massachusetts Breast Cancer Coalition. And I want to welcome you to today's important discussion. I am pleased to welcome today's speaker, Catherine Rogers. Catherine is a staff scientist at Silent Spring Institute. She joined Silent Spring's team in 2012 and has worked on a number of studies researching exposures to endocrine disrupting chemicals in water, indoor dust, and indoor air. She is currently leading a study that includes several universities to examine and reduce chemicals of concern on college campuses as part of the Healthy Green Campus Project. Catherine is motivated by Silent Spring Institute's commitment to community-based participatory research and to advancing exposure reduction strategies. She has a master's in public health from Boston University with training in toxicology and risk assessment. Catherine, welcome.
Thank you very much, Michelle. I'm so pleased to be on today's webinar, and thank you to MBCC for inviting me to present this. Today I'm going to be talking about a new review that Silent Spring Institute just conducted, looking at environmental chemicals and breast cancer. Oh, my slide's not advancing.
The broadcast is now starting. All attendees are in listen-only mode.
Okay, now the slide's advanced. So just to start, I wanted to go ahead and say that this review, and actually all of Silent Spring Institute's work, is done under a prevention science framework. And what that really means is, if we're going to look at the biological activities of chemicals and understand their pathways to breast cancer, and then we're going to look and see how are people exposed to those chemicals. Are exposures widespread? Are there certain people who have unusually high exposures? That leads us to take action, either to educate, regulate, or reformulate. And so that really is the guiding principle of Silent Spring's research and approach to science. So here's just a screen grab from this new paper that we published in environmental research, and we're calling this, it's an updated review of epidemiological literature informed by biological mechanisms. So basically we looked at chemicals and human studies of breast cancer, and we reviewed a total of 158 articles over a 10-year period in this paper. And the goals of our review were to see what we could understand from unique exposure scenarios. So like women who have unusually high exposures, or I'll talk a little bit later about some of these studies that allow us to really get a better understanding of what happens when there's a defined exposure window and exposure level among women. We also wanted to help create a guide for future epidemiological studies. If there are going to be studies going forward with looking at the human outcomes of breast cancer, what's the best way to design those studies? We wanted to be able to answer that question. And then another big goal of this review was to compare the biological activities of the chemicals investigated in these studies to how the studies were designed. Were they designed to capture the biological activity of the chemical in question? And we also wanted to write this in a way to speak to skeptics, so we could really pull out what the strongest evidence is. So for anyone who is unfamiliar with this, or who doesn't think that there is any link between environmental chemicals and breast cancer, to really make the strongest case for what we do know. So this review, it has the word update in its title, because this is actually an update from an epidemiological review that was published in 2007 by Silent Spring Institute. It was also accompanied by a review of chemicals, 216 chemicals that are known to cause mammary gland tumors in rodent studies. So that has really, since 2007, has been a roadmap for future studies examining risk from chemicals associated with breast cancer, both identifying what those chemicals are for laboratory studies and then also for human studies. So this was an update on this 2007 publication. And in 2007, the review that we published had positive findings for exposure to PAHs. Those are chemicals from combustion that can be found in car exhaust, from PCBs, industrial PCB chemicals, and some gene interactions, and some associations for exposure to organic solvents and dioxins. We did find some limitations of the studies that we reviewed in 2007, which were that often studies were designed to ask the wrong question and they did that over and over again. So for example, collecting blood from at diagnosis from older women and examining that blood for DDE exposure, which is not I'll talk more about this later, but if you wanted to look at DDT exposure, looking at DDE is not a great proxy for exposure to DDT earlier in life. So those studies were showing up to be negative with good reason. They really weren't designed very well. And another limitation in the 2007 review is that a lot of the studies include populations that didn't have a high exposure. So there weren't very good comparison groups. And just to see what has happened since 2007, if you go online and you look at major breast cancer organizations to try and understand, well, how do people account for environmental risk factors in breast cancer? This is an example from the Komen website. They are, for this example that you see on the screen of the pesticide DDE and PCBs, don't increase the risk of breast cancer. These are examples of actually findings from studies that weren't well designed. So again, this is one of the goals of this review was to speak to the skeptics and really say, well, what do we know from the strongest evidence that we have at this point? How can this be, and how can these more accepted rules of breast cancer risk factors incorporate the risk from environmental chemicals? So our methods for this review are that we searched the literature, peer-reviewed literature for over 130 chemicals and breast cancer. And these are chemicals or chemical groupings that were pulled from our mammary carcinogen list that was published in 2007. These are chemicals that were also that also have widespread exposure. What we did was we summarized the breast cancer biology of these chemicals basically by looking to other established literature such as the International Agency for Research on Cancer and the National Toxicology Program, as well as published literature to ask some basic questions about how are these chemicals associated with, what do we know about their biological activity that's associated with breast cancer? And then, like I already said, we actually reviewed 158 studies that looked at human breast cancer. So we went into this review with a little bit of knowledge. I'll just try and share with you quickly. But basically this is recognizing the fact that breast cancer is a complex disease, and it's not even just one disease. And we wanted to, in understanding in matching the biological activity of the chemicals being investigated to how studies were designed, we wanted to look through biological pathways to breast cancer. We wanted to look through that lens when we were assessing how well these studies were designed. So just quickly, I'm going to talk about a few biological pathways to breast cancer. The first one is DNA damage. So this can be thought of for ionizing radiation, which can cause double strand breaks in DNA, as an example. Also chemicals that can act as endocrine disruptors or tumor promoters. An example of that is hormone replacement therapy that can cause estrogen-dependent cell proliferation. And another example are chemicals that can act as endocrine disruptors that disrupt the development of the mammary gland, that can actually make it more susceptible to a carcinogen later in life. And an example of that is the drug DES. So there's also some similarities of established breast cancer risk factors and some of our chemicals of interest. So for example, I've talked about on the left, you'll see some established breast cancer risk factors. And so reproductive factors and prescription hormones, the way that they can act as far as being endocrine disruptors or tumor promoters, we also see some similarities to this with environmental and endocrine disrupting chemicals. Alcohol is a known risk factor for breast cancer. It can lead to tumor promotion and increased levels of estrogen in the body. And solvents, I mean, alcohol is a type of solvent, and there are other solvents that act in a similar pathway to ethanol or alcohol that we drink. I gave ionizing radiation as an example of DNA damage. There are also other chemical genotoxicants, which I'll present in a few slides. And then tobacco smoke is also a known risk factor for breast cancer. And PAHs, or products of combustion, also contain many similar chemicals as to what can be found in tobacco smoke. And I also just wanted to recognize that within these pathways, or within these similarities of environmental chemicals and the known risk factors for breast cancer, there's a little bit more nuance to it as well. So this is actually from, this table right here is from a paper that Silent Spring published with collaborators on a project called the Pathways to Breast Cancer Project. This is trying to better understand how we can screen chemicals for breast cancer. But basically, if you look at this top row right here, looking at cellular and molecular events, you'll see, for example, genotoxicity listed. Well, it's not as simple as just thinking about the genotoxicity of a chemical, because there's also other biological factors that come into play, such as the DNA ability to repair DNA damage, or the ability to alter apoptosis, excuse me, or cell death. Or immune modulation. So there's different types of biological factors or events that happen in conjunction with environmental exposures that can alter risk. There's also different biological, tissue changes or other biological factors about someone's body, including breast density, is becoming increasingly it's an area of research as a risk factor for breast cancer. And then this goes along with understanding how endocrine disrupting chemicals can disrupt the development of the mammary gland. How do certain chemicals change breast density with early in life exposures? And could that be a pathway to breast cancer later on? And then, of course, there's other susceptibility factors, including genetic polymorphisms and other things, including the early onset of breast development or early puberty and lifetime exposure to estrogen. So in addition to coming at this review with the lens of understanding what are the pathways to breast cancer and how do environmental chemicals fit into those, we also have to understand that there's other biological factors within the body that can modulate these risk factors as well. And this basically is just what I've just said, what I've just talked about. Breast cancer is a very complex disease, and in addition to all the things I've just talked about, there are there's susceptible windows of development, of breast development, that depending on the chemical can influence the risk of breast cancer as well. So those periods include the time before birth, when the mammary pad is first developing in utero. During puberty, the first time when those breast cells are differentiating and proliferating into what their final form will be, and during a woman's first pregnancy, when they finish that differentiation and proliferation, and then during menopause, when the breast can be more susceptible to tumor promoting chemicals. So this is the framework for our review, and this is a figure that's meant to take into account not all of the factors that I've just talked about, but some of them. And so I'll walk through this figure. So in the leftmost column, sorry, you can see there's 16 categories of chemicals that we investigated. And for each of those, we looked at their biological activity related to breast cancer, like I described earlier. So that column G is for genotoxicity. If there's a check mark there, that indicates that there is evidence of genotoxicity. Under that column that says RMC, that's rodent mammary carcinogen. A check mark there indicates that a 2-year bioassay in adult rodents has found evidence of mammary tumors. Then that's adult rodents only, because that's often, well, that's what the standard protocol is. For EA, that is endocrine active, if that chemical is endocrine active. So that could be causing cell proliferation, estrogen-dependent cell proliferation, or progesterone cell-dependent proliferation isn't a standard outcome that's researched, but it could include progesterone, binding to the progesterone receptor as well. Now, moving over to the right, you can see this this larger box on the top called exposure window. And so this is setting us up to look at, were the human studies looked at the exposure window. And what these rodents are showing is there animal evidence of changes to the mammary gland during these certain exposure windows. And so you can see that in utero and in childhood and adolescence, there is some evidence of changes to the mammary gland following exposure to those chemicals during those exposure windows. So hopefully this helps kind of lay out the framework of which we conducted this review. And here is one of the major findings. Before I delve in, we're going to go through each of these chemical classes. But before I do that, I just want to show you an interesting finding from our study, which was that we looked at the number of studies that included whose study design included looking at specific exposure windows. So you can see the color gradation here in this figure shows that the gray means that there are 0 studies that looked during this exposure window, to 1 to 2, so on. And what's interesting is if you look at the periods of breast development in utero, childhood, and adolescence, and before pregnancy, you can see that there is a lot of studies miss this critical timing. They miss looking at these exposure windows for which we actually do have laboratory evidence of altered mammary gland development. A couple of them do, but a lot of studies are not designed to look at very specific windows of development. And actually, if you look over to the rightmost column, the non-specific column, you can see most studies fall into that category where they're not specifically looking at any particular window of breast development. But you can see that there actually are more studies that fall within this premenopausal exposure window. So this is a slide that has another layer here, and what I've done is I've just added hashing over the cells within this period of breast development and premenopausal exposures before diagnosis, for those exposure windows that had positive findings. So not only does here to show what the papers that we reviewed looked for, but then also is now showing their findings. And so you can see there are a number of hashed cells where there is not a mouse, where there isn't a biological evidence of mammary gland changes, but some of them are. So we can also, we're learning new information from these studies as well that can help inform what we already know about some of these chemicals from lab studies. So first, so now I'm going to use this figure as a guide to help us walk through the results from the review that we conducted. And you can see first here, I'm going to talk about a class of chemicals, persistent endocrine disrupting chemicals. So that includes dioxins, PCBs, organochlorine pesticides, and then I'll talk a little bit more about what xenoestrogen burden means. So first, to talk about dioxins. This is one of those examples of a unique exposure scenario in Italy in 1976. There, unfortunately, at a chemical manufacturing facility, there was an explosion that led to very high levels of dioxins in the surrounding area. And those women who lived in that area were enrolled in the Seveso Women's Health Study and followed for a number of years, actually up to 30 years. And what we've learned from this study is that the women who had the highest blood levels of TCDD, which is the most potent dioxin, had more than a 2 times greater increased risk of breast cancer after 11 to 20 years of follow-up. The risk did not, at the time point of 30 years of follow-up, the risk wasn't as high. So that shows that the exposure window for the effects of TCDD might actually be earlier. But as these women continue to be followed, we'll find out more about them, about what the longer term effects of TCDD exposure could mean. So now I'm going to talk about organochlorine pesticides. This was a really great study that we included in our review called the Child Health and Development Study. And this included women in California. It collected blood samples from these women right after they had given birth from 1959 to 67. And this was another unique exposure scenario because the blood was collected from these women during years of peak DDT use, before DDT was banned. And what we found from the mothers was that for those women who were younger than age 14 in 1945, when DDT first came on the market, was first used in the United States, they had 5 times the higher odds of developing breast cancer later in life, before age 50. And the offspring, the female daughters from those women are also included in the study. And it was found for the women who had higher levels of DDT in their blood when during that time right after they had given birth, those women who had those higher levels meant that those daughters in utero also had those higher levels, and they actually had almost 4 times higher the risk, or 3.8 times the risk of developing breast cancer before age 50. So we can see increased risk in the mothers and then also in their daughters. There were 2 other studies that looked at exposure to DDT in a different way, which is very interesting. In case, it could be very hard to, or impossible to replicate that study of being able to collect blood from women at that point in time. But instead, other studies asked a question to women: did you ever see a Foggart truck as a kid? Which is interesting because it's quite a memorable experience. And the Long Island breast cancer study found an increased risk in premenopausal breast cancer for women who did see a fogger truck as a kid. And the prospective sister study found an increased risk of estrogen positive, progesterone positive receptor tumors in those women who had seen fogger trucks as a kid. So now I'm going to talk just briefly about PCBs, or this is a group of industrial chemicals, and it's a diverse group. There's over 209 individual congeners that fit into this grouping. They're used in circuit boards and as insulators up until the 1970s. So what I'm showing you here is that if you look at the left column of individual studies, and then you look across the top column labels, basically there are many different PCBs. They can be grouped into different biological activities. And so many studies did look at groupings of PCBs, looking at PCBs that are potentially estrogenic, those that are dioxin-like, those that are persistent, and those that are CYP-inducing PCBs. And because the studies didn't have perfect overlap with how they grouped these chemicals or what they had available in blood samples, it was hard to tell what the trend really was. There were mixed results. Some studies found an increased risk of breast cancer, and some found a decreased risk of breast cancer. However, that child health and development study that I talked about with the DDT also looked for PCBs, and they found an increased risk with one of the CYP-inducing PCB chemicals, 203. And interestingly, that study was not replicated in other studies that looked for 203. So this right here is just a plot showing the results from the child health and development studies. So you can look and see what the difference in the risk of breast cancer is in the mothers who had high DDT exposure, those who had high DDT exposures before they were 14 in 1945 when DDT came into use, the high DDT levels in their daughters, and then high PCBs, really that PCB congener 203 in mothers. So if we go back just to look at our framework really quickly, you can see where there's positive findings that overlap with what rodent studies have shown for organochlorine pesticides, and what we've learned from dioxins. So the study that looked at the Seveso women's health cohort enrolled women who were aged 0 to 40. So that hashed cell showing the exposure window as being premenopausal before diagnosis does include, probably because the women were age 0 to 40, does include likely women in those other periods of breast development, but those just aren't highlighted because it wasn't, the study wasn't specifically designed to look at those individually. So next I'll talk about non-organochlorine pesticides, or unspecified pesticide use or exposure. And there were a number of studies that found increased breast cancer risk with non-specified pesticide use. For example, lifetime application of indoor pesticides or lawn pesticides, the use of insect repellents. Some studies did look at exposures earlier in life. So for example, this sister study asked women if they had lived on a cotton or a tobacco farm before age 18 and found an increased risk for those women. And women who lived near a farm and saw pesticide drift before age 20 were also shown to have increased breast cancer risk later in life. And a few of the studies, and really one in particular, did a good job of looking at non-organochlorine pesticide use, so looking at chlorpyrifos or terfubos, sorry about that, was from the agricultural health study, and that was looking at actually the female spouses of professional pesticide applicators, asking about if they use pesticides at home. And so that, I think that was the first study to really show an increased risk of breast cancer from non-organochlorine pesticides. And I'm sure that there will be more studies coming out looking at, specifically looking at non-organochlorine pesticide use. You can see here positive findings for early life exposure to pesticides, which is consistent with what animal evidence has shown us as well. So next I'm going to talk about PAH biomarkers and air pollution. So PAHs are these chemicals formed from combustion, from burning, or from car exhaust, and they can have many, or they are one of many components in air pollution. So the Long Island breast cancer study investigated exposure to PAH DNA adducts and also looked at a number of genetic polymorphisms. And you can see here on this plot, over here on the left with the odds ratio of 1, all of the boxes that are above 1 indicate an increased risk of breast cancer. And I've grouped the types of genetic polymorphisms down on the bottom. So you can see it's interesting where these, there's a cluster of increased breast cancer risk for genetic polymorphisms in DNA repair genes. So that may mean that if there's a PAH DNA adduct, that could lead to genotoxicity. If there is a deficient DNA repair system, then that could in fact, or that is associated with higher risk of breast cancer. So this is an interesting area of study. This is really just a handful of the genes that could be involved in this pathway, but this was a new area that has been more thoroughly investigated since our review in 2007. And air pollution, which is now a known human carcinogen, designated by the International Agency for Research on Cancer, was comprised nearly, or over 25% of all of the studies included in our review were looking at air pollution. So there's a huge increased interest in looking at air pollution as a risk factor for breast cancer. And studies used many different exposure proxies for air pollution. So carbon dioxide, particulate matter, NOx and SOx, and the studies that found, and so the studies that looked at this, many studies did find an increased risk of breast cancer, although not all did. And I think again, it's because one reason might be because different exposure sources were included across all of these studies. But the studies that did find an increased risk of breast cancer stratified by menopausal status and often found increased risk of breast cancer in postmenopausal women. Some looked at tumor subtypes. So for example, the sister study found increased risk of estrogen receptor positive breast cancer with exposure to air pollution from car exhaust. And some studies included or looked at traffic exposure earlier in life, so say between age 18 and 30. And those also found an increased risk of breast cancer in postmenopausal women. So this really just gets back to the fact that there are many different components of air pollution, and also that breast cancer is a complex disease. So we're able to see differences in risk for those studies that looked more carefully at a premenopausal disease or a postmenopausal disease and different tumor subtypes and also different exposure windows. So I imagine that the focus on air pollution and breast cancer will continue to be a big area in breast cancer research. This is just bringing us back to our framework to see that there were indeed positive findings for air pollution during windows of breast development. So next I'm going to talk about a group of chemicals that I've called consumer product chemicals. So this includes BPA, phthalates, nonylphenols, hair dyes and straighteners, PFAS chemicals or highly fluorinated chemicals used in stain-resistant or water-resistant coatings, and flame retardants. And so we do have some evidence of, if you look at the column of breast development, you can see those rodents there. We do have evidence of mammary gland disruption for a number of these chemicals. Oh my gosh, it shouldn't look like that. Sorry about that. So there were a number of issues with the studies that include consumer product chemicals. Many of these chemicals are rapidly metabolized and have a short half-life. So a one-time measurement may not be representative of someone's overall exposure. And the timing of exposure remains an issue as well. So collecting a urine sample from someone after breast cancer diagnosis may not really tell you about what their exposure was like a week ago, or nonetheless during windows of breast development. So we did see there were definitely an increased number of studies that examined exposure to consumer product chemicals. And I think that in the future they will continue, the study designs will continue to get to improve to better look at this. Some of the things that we did find was that there was an increased association with breast cancer with lifetime use of household cleaners and air fresheners. Based on other studies that Silent Spring has done looking at the types of chemicals that are found in household cleaners and air fresheners, that can include phthalates and nonylphenols, which are also included in our list of chemicals. We did find that male breast cancer was associated with occupational exposure to alkylphenols. So we may be exposed to consumer product chemicals as consumers, but there's also people who are working with these chemicals in a more industrial setting who have exposures to these chemicals. So it will be important to look at those populations as well, because their exposure levels are likely higher than a consumer's exposure. Probably the most interesting finding from this group of consumer product chemicals studies was from the Danish National Birth Cohort, which enrolled 100,000 pregnant women in 1996 and followed them to 2010. They're still being followed now, but the data that we have is when they're followed to 2010. And it found that women who had higher levels of PFOSA, which is a precursor chemical to PFAS, a PFC or a highly fluorinated chemical, had higher risk of developing breast cancer 10 to 15 years later. And this is interesting because this is one of the first studies that had a prospective design that was able to follow women over time after being exposed to a chemical that can disrupt mammary development and is endocrine disrupting chemical during a sensitive window of breast development. So this is another unique cohort that has current exposures, exposure levels for chemicals currently in use. So if we bring this back to our framework here, we can see that there are positive findings for these consumer product chemicals. For the few studies, you'll see there's only 3 studies that actually looked specifically at windows of breast development, or that would incorporate windows of breast development. I'll talk briefly about studies we included of drinking water. We included drinking water, even though that's not a specific chemical category. Drinking water can studies by Silent Spring and others has shown that drinking water can be contaminated with consumer product chemicals, pharmaceuticals, and this can happen through effluent or wastewater contamination into the drinking water supply. And there's also other studies of drinking water that have been contaminated with solvents that I'll talk about in the solvent section. But these drinking water studies use nitrate as a proxy for wastewater contamination, or look at historical use of a specific drinking water source. So within this group, there were 2 studies that were conducted on Cape Cod, and one found a slight increase in breast cancer risk for women who were in 5 districts where drinking water was contaminated by wastewater. And another study conducted by Silent Spring Institute used average nitrate levels per water district and didn't find a significantly increased risk. So maybe one limitation of these studies are historical reconstruction of water, and people may have other drinking water sources, and using an average nitrate level may not necessarily capture all the variability within a drinking water district. But I thought those were 2 interesting studies. 2 other studies that looked at nitrate levels as a marker for wastewater. One of them did find, that was conducted in Spain, actually found a non-significant increased risk of breast cancer for exposure to nitrate in drinking water in early adulthood, before I think age 20 to 30. And another study that was conducted in Iowa, in the Iowa Women's Health Study, that looked at daily nitrate exposures, found a non-significant increased risk of breast cancer as well. So drinking water, I think, is another interesting area of study, although I think studies looking at disinfection byproducts or other specific contaminants of drinking water were lacking. And it would be good to see for future studies to see more focus on some specific contaminants of drinking water, including PFAS chemicals. So lastly, I'm going to talk about exposures to organic solvents and women's occupation. And coming from our 2007 review, there was some suggestive evidence of increased risk of breast cancer from organic solvents in our occupations. But one of the big limitations that we had identified in our 2007 review was a lack of highly exposed populations. And a number of occupational studies did not include breast cancer, did not have enough women to have the power to calculate increased risk of breast cancer. So I've put this slide together talking about solvents in occupation, but I'll talk about them a little bit separately. First, I'm going to talk about solvents. So the prospective sister study did find increased risks of breast cancer with increased duration of solvent work or an occupation involving solvent use before a woman's first childbirth. So this is looking at occupational solvent exposures during periods of breast development. And this was also found in another study that looked at any type of solvent work, so not considering duration, but any type of solvent work before a woman's first childbirth, and finally looking at solvent exposure before age 36. So all 3 of those studies that are looking at occupational solvent use during windows of breast development found increased risk. And there were a couple of unique exposure scenario studies that were in this group that basically were able to look at the risk of breast cancer following worker protection rules. So for example, one study that was conducted in Taiwan found that women who were exposed to TCE used in the facility before it was removed from the facility in 1974 had a significantly higher risk of breast cancer compared to the women who worked after 1974 when TCE was removed from the facility. This was also seen in the United States in the sister study comparing women who worked with solvents before and after 1980, when there were many restrictions put on solvents in the United States. So those are some specific studies looking at solvent use, occupational solvent use. And then there were also many studies that were broad occupational surveys. And so I'm going to just summarize those quickly here on this slide. So there were a number of occupations that potentially had exposures to chemicals that would be biologically related to breast cancer. And those included work in the auto industry, work with plastics, work with rubber manufacturing, and in textiles and in healthcare work. So these studies didn't get down into what a woman's individual level exposures were, but more looked at women who had worked in these industries, what their breast cancer risk was. One of the common limitations of occupational studies is including the appropriate reference group, because women who work in jobs that have a higher degree of physical activity, which can also be associated with blue-collar jobs. Because physical activity is protective of breast cancer, those women may have a reduced risk of breast cancer. So if you're comparing women who have a high degree of physical activity to the risk of women who don't necessarily have that high degree of physical activity, you can introduce something that's called the healthy worker effect. So the good studies that were included in our review, including the sister study, actually used an appropriate comparison group, but not all of the studies did this. And the strongest studies that we reviewed considered the intensity and the duration and the timing of solvent exposure. And so you can see down here that there were positive findings for exposures to solvents during these windows of breast development. And while it's true, if you're looking at the right side of my slide, there were positive findings for postmenopausal and non-specific and lifetime exposures to organic solvents in occupation. And I think I just talked about why that was. So the conclusions for a study for our review is that, like I said, one of the goals was to really pull out what the strongest evidence was, what the strongest studies were. And one of our biggest conclusions is there are some really great studies that have been done that prospectively look at a woman's exposure and subsequent risk to breast cancer, and we should rely on those strong studies. And we should support those cohorts. The Child Health and Development Studies now is following 3 generations of women. And the Danish National Birth Cohort Study is also another unique cohort that collected blood during women's pregnancy. And we want to think more about what happens before a woman develops breast cancer. So this is a suggestion for future studies. So what I was talking about at the very beginning, where there's biological factors that can also influence the risk of breast cancer, including breast density. Well, is breast density affected by chemical exposure? Is it really all in the same continuum? So we are conducting a study with girls in Santiago, Chile, looking at environmental chemicals and risk of breast density during puberty. More studies should consider, in the future, should consider mixtures and interactions. There were very few studies, but we did include a couple in our review that looked at xenoestrogen burden, or really what the mix of estrogenic effects are from a chemical. So not looking at one chemical in particular, but saying what is the risk from an overall increased level of exogenous estrogen. So we're exposed to many of these chemicals at the same time. It's not like we're exposed only to one chemical at a time. So there are limitations looking at exposures to chemicals individually, and we need to better be able to look at risks from exposure to mixtures of chemicals. And then lastly, we need to be able to develop rapid tests to predict breast cancer effects, to predict how the levels of proteins change could lead to breast cancer, DNA damage. And so we do have a project at Silent Spring called BC Screen that's aiming to do that. And I just want to show this slide. This basically just shows the concordance between animal evidence and human evidence. So you can look at, on the left column, you can look at the type of exposure, and you can look to see what we know about its association with human breast cancer and with rodent mammary tumors. And so in general, we can see that there's a fair amount of agreement between what we know from rodent mammary tumors and what we know about human breast cancer. And we can continue to use rodent mammary chemicals that are rodent mammary tumors as predictors of breast cancer. And I'd like to thank my review authors, which include Julia Udesky, Ruthann Rudell, and Julia Brody at Silent Spring Institute. And the study is freely available on our website. And a lot of the tips, if you want to know what you can do, a lot of tips that we recommend to reduce exposures to environmental chemicals associated with breast cancer are included in our free app called Detox Me. Right, thank you very much. So I think hopefully I've ended with enough time for some questions.
Wonderful, thank you, Catherine. I've got a couple that have come in. One, in an earlier slide in one of your groupings, you had apoptosis. I hope I'm pronouncing that correctly. Mm-hmm. And the question was, what is that? Could you explain what that is?
Yes, apoptosis is a programmed cell death. That's a natural thing that happens in our bodies. It can also be a body's defense mechanism to prevent cancer. If there is a cell that has a change in it, our body has a natural process called apoptosis, which is coded by a certain set of genes that we have, basically, that can then get rid of that cell. If we aren't able to regulate the growth of our cells, if we have a deficient gene that can regulate apoptosis, that could be a risk factor for any type of cancer.
Okay, thank you. Another question that's come in is, and I'm not sure, this is a rather broad question, but I'm really curious to hear your answer, is how do you avoid another DTT situation?
That's a great question. So DDT being the chemical that women who were exposed to that chemical when they were younger than age 14 in 1945 had a 5 times higher risk of breast cancer later in life. So I think that if we looked at the activity of DDT, and in fact, if we look at the biological activity of DDT now, we can understand that maybe we would expect to see that effect. It disrupts the development of mammary tissue, and it's an endocrine disrupting compound. It has some estrogenic properties to it. And one thing that we can do now is we can use what we have, the knowledge that we have about from laboratory studies and from animal studies, showing these types of effects to use our precautionary principle that I showed, that was the first slide that I showed, that said, if we have biological evidence of toxicity or of evidence of a pathway to breast cancer, and we have widespread exposure, we have enough information to take action. So there are a number of those chemicals included in the consumer product chemical grouping, where right now we don't have a lot of very good epidemiological studies on those chemicals, but we do have a fair amount of laboratory and rodent studies on those chemicals. And so you'll see if you look at our Detox Me app, you'll see that we do actually recommend avoiding a number of those chemicals, even without having all of the necessary human evidence to prove that. We're acting on the precautionary principle.
Okay, thank you.
Mm-hmm.
I've got a couple of questions that have come in on the same point, so I think it was missed earlier. On the graph that you had, could you explain what RMC stood for?
Oh, sure. That is a column that we checked off for any chemical that tested positive for a rodent mammary gland carcinogen. And that was determined from studies of adult rodents that were exposed to a chemical as adults, and then over a 2-year time period, if they developed mammary tumors, then that chemical would have a check mark in that column.
Great,
so
it's
interesting because it is the standard way to do a tumor study in animals is to expose adult animals, whereas it doesn't fully capture the all the pathways to breast cancer that include exposures earlier in life, from in utero to, through puberty, through a woman's first pregnancy.
Okay, thank you. I've got one more for you.
Okay.
How does Silent Spring follow products that had endocrine disruptors years ago to confirm whether those products are still having those harmful effects?
So I'll try and answer that. So in 2012, Silent Spring published a study where we tested about 150 consumer products bought off the shelf for 66 endocrine disrupting compounds. And we found that there were a number of, I think we found at least 50 of those endocrine disrupting chemicals in the products that we tested. And that was a snapshot that allowed us to see the prevalence of these chemicals in consumer products. It also allowed us to see which chemicals were included on ingredient labels and which ones weren't. There are a number of chemicals that are not included on ingredient labels, which is why labeling is such an important thing to advocate for. And the limitation of that study is that it was a snapshot. To test those products for our suite of chemicals is hundreds of dollars per test. So we don't have a program to continually test products, but we do have information about the types of chemicals that are expected to be in these types of consumer products, as well as information about which ones you can avoid by looking at ingredients labels and which ones are harder to avoid by looking at ingredients labels. And our Detox Me app it's not really a product-based app the way that the Environmental Working Group has Skin Deep, where they rate the ingredients of products and give an overall rating to a product. But we do provide more tips that aren't necessarily purchasing based, but things like keeping dust levels low in your home is a good way to reduce exposures to flame retardant chemicals, for example, as well as avoiding cleaning products with disinfectants in them, for example. But it's meant to be more broad than just specifically looking at individual products.
Great. Well, thank you, Catherine.
You're welcome.
This has been a great presentation. You've provided us with so much information, so much to take in. On behalf of Cheryl Osimo, NBCC's Executive Director, and our Board of Directors, I want to thank you for giving us this presentation today. and for the information that you've provided. And I want to thank those of you that have attended for joining us this afternoon. And I appreciate you taking the time to be a part of the discussion. And I want to wish you all, thank you. And I want to wish you all a very happy afternoon.
Thanks.